friends, welcome to entranceadvisor.com. Today in this video I am going to explain about the fertilization and implantation of the human reproduction. Before that, if you are new to my channel, let's hit the bell icon and do subscribe to my channel. So let's start. Before that, you need to know about some terms. What is insemination? During the coitus or copulation, the semen is released into the vagina or the birth canal by the penis. The motile sperm swim rapidly through the cervix, enter into the uterus, and finally reach the ampullary region of the fallopian tube. The ovum released by the ovary is also transferred to the ampullary region of the fallopian tube. The fertilization occurs in the fallopian tube only if the ovum and the sperms are simultaneously transferred into the ampullary region of the fallopian tube. What is fertilization? It is the fusion of the sperm and the egg. During the fertilization, the sperm induces changes in the zona pellucida layer of the ovum that block the entry of the additional sperms, ensuring that only one sperm can fertilize an ovum. The secretions of the acrosome help the sperm to enter the ovum through the zona pellucida and the plasma membrane. This induces the secondary oocyte to complete the meiosis. This is again an unequal division. It results in the formation of a second polar body and a haploid ovum. The haploid nuclei of the sperm and the ovum fuse to form a diploid zygote. The zygote contains 46 chromosomes, that is 2n. The sex of the fetus is determined by the sex chromosome present in the sperm. As the female is XX, the ovum will always carry the X chromosome. The males are XY, and therefore the sperm can contain either X or Y. Therefore, half of all the sperms carry the X chromosome and other half carry the Y chromosome. Depending on whether the X containing the sperm or the Y chromosome fuses with the ovum, the zygote will be female or male. The zygote undergoes mitotic cleavage as it moves along the isthmus of the oviduct towards the uterus. It forms 2, 4, 8, 16 daughter cells called as blastomeres. What is morula? The embryo with 8 to 16 cells are called blastomeres. And the morula continues division as it moves further along into the uterus. The blastomeres arrange it into an outer layer called the trophoblast. The inner cell mass is attached to the trophoblast. The trophoblast attaches to an endometrium. The inner cell mass differentiates to form the embryo. The cells of the uterus divide rapidly and cover the blastocyst. The blastocyst thus embeds in the uterine wall. This is called the implantation. This leads to the pregnancy. So what is fertilization? Let us have a look in a detail. The fertilization is the fusion of the haploid gametes, egg and the sperm to form the diploid zygote. Note, though there can be a subtle differences in the fertilization process which occurs naturally within the body or through reproductive technologies outside the body, the overall product in both cases is a diploid zygote. In fertilization research, after humans, the mouse is the most studied species, followed by the domestic and farm animals. The process of fertilization involves the components of and signaling between both sperm, spermatozoa, and egg oocyte. So, fertilization in human refers to the fusion of the male and female gametes that facilitates the development of the new organism. So, here you can see the journey of an egg. Step 1 is the egg leaves the ovary and enters the fallopian tube. Okay. In the steps 2, the sperm enters the egg and unites with the nucleus. Then in the step 3, the fertilized egg divides 2 cells, then 4 cells, then 8 cells. These are called blastomeres. And in the uterus, the cells attach to the uterus in the last step. Here you can see the fertilization is a generalized acrosomal process where the sperm makes a contact with an egg. The acrosome reacts with the zona pellucida. The acrosome reacts with the perivitelline space, perivitelline space that is, and the plasma membrane of the sperm and egg fuse. The sperm nucleus enters egg and the cortical granules fuse with the egg plasma membrane, which renders the vitelline layer impenetrable to the other sperms. Okay? And this is the structure of the ovary. This layer is called zona pellucida. This is perivitelline space. This is, and this is egg plasma membrane. These are cortical granules. This is sperm nucleus, and this is vitelline layer, and this is egg cytoplasm. So these are more pictures so that you can understand more clearly. Hundreds of sperms attach to the corona radiata, which is the outermost layer, and begin to break through the barrier of the granulosa cells and approach the zona pellucida. Then they contract with the zona pellucida, then triggers the acrosome reaction, causing the sperm to secrete digestive enzymes that break down the glycoprotein membrane of the zona pellucida and help to expose the oocyte's plasma membrane. Then the single sperm succeeds in burrowing through the corona radiata and zona pellucida and making contact with the oocyte's plasma membrane. The sperm's plasma membrane fuses with that of the oocyte and the sperm releases its nucleus into the cytoplasm of the oocyte. The vitelline layer is this, I have already said, this is jelly coat and this are acrosome reaction is taking place. These are fused plasma membrane and this is the cortical granule contain. Okay, and you know the structure of the sperm, that this is a sperm head, this is mitochondrial material, and a nucleus is present, actin is present, and acrosomal granule is also present. So an egg and the sperm forms a diploid zygote. Now the steps of fertilization in humans. The fertilization process in humans takes place in several stages involving both the chemical and physical events. The different stages of fertilization in humans are mentioned below. First of all, the acrosomal reaction. This is acrosomal reaction. The sperms in capitation undergo the acrosomal reactions and release certain chemicals known as sperm lysins, okay, present in the acrosome. Due to the acrosomal reactions, the plasma membrane of the secondary oocyte and the sperm fuse together so that the contents of the sperms can enter. When the plasma membrane of the sperm binds with that of the secondary oocyte, the plasma membrane of the oocyte depolarizes. This prevents the polyspermy. No other sperm can't enter. And calcium ions play a significant role in the acrosomal reactions. As the main factors essential for acrosomal reactions are optimum pH, temperature, and calcium and magnesium concentration. And then the cortical reaction. The cortical reactions is another reaction. This process is cortical reactions. When soon after the fusion of the plasma membranes, the oocyte shows cortical reactions. The cortical granules found under the plasma membrane of the oocyte, which fuses with the plasma membrane and releases the cortical enzymes between the zona pellucida and the plasma membrane. The zona pellucida is hardened by the cortical enzymes and prevent the polyspermy. As I said, no other sperm can enter. And the sperm entry, then this entry of the sperm nucleus, a projection known as the cone of reception is formed by the secondary oocyte at the point of the sperm contact. This cone of reception receives the sperm. Okay, so first of all the contact, then the acrosomal reaction, then the growth of the acrosomal process, then fusion of the plasma membrane, then the entry of the sperm, and then at last the cortical reaction. Now the karyogamy. After the entry of the sperm, the suspended second meiotic division is completed by the secondary oocyte, as I said in the previous lecture. This gives rise to a haploid ovum and the second polar body. The head of the sperm containing the nucleus detaches from the entire sperm and is known as the male pronucleus. The tail and the second polar body degenerates. The nucleus of the ovum is known as the female pronuclei. The male and the female pronuclei fuses and their nuclear membranes degenerate. Then the fusion of the chromosomes of the male and the female gametes is called the karyogamy. The ovum is now fertilized and is known as a zygote. So you got it? The fusion of chromosomes of male and female gametes is known as karyogamy. Gamy means mating or marriage. Okay. Now the activation of the eggs. The entry of the sperm triggers the metabolism in the zygote. Consequently, the protein synthesis and the cellular respiration increases. Then implantation occurs. Once the fertilization happens, the cell starts to divide and multiply within the 24 hours in the fallopian tube. This detached multicelled structure is called the zygote. Later, after 3 to 4 days, it travels to the uterus and now we call it as an embryo. When it is attached in the uterine wall, we call it an embryo. Okay? The embryo develops and undergo various stages and gets attached to the endometrial layer of the uterus. This process of attachment is known as implantation. The process of attaching is known as implantation, and the cells, the lump of cells which gets attached to the uterus is known as embryo. You understood? Now the sex determination. How a sex is determined? And the fertilization is a process in which a new cell is formed when two gametes, the sex cells, the sperm and ova fuse together. So during this unbiased event, all the genetic information is transferred from parents to the child and the gender of the child is determined. So further determines the sex of the child, that is, if the sperm carries an Y chromosome, the child will be a boy, and if the sperm carries an X chromosome, the child will be a girl. In this picture, it is clear that the female sex organ contains XX and the cells in the testes are XY. So, if from the sperm cell, if X comes, then it is female, and if Y comes, then it is male. It's just simple. And this is another detailed process. I have earlier described this. The first of all, the sperm contacts with the egg, and then a chromosomal reaction takes place, and through a series of process, then the plasma membrane and the sperm fuses, and the sperm nucleus and the egg enters, and then cortical reactions takes place by fusion with the egg plasma membrane, which renders the vitelline layer impenetrable to other sperms. Okay. Now the implantation. When the zygote moves through the isthmus towards the new uterus, it undergoes mitosis. Number of cell divisions occurs repeated and fast. The cell divisions in the zygote are called the cleavage. The process of division, the cells which divides, you can see in this picture that in this here, one from one cell to two cells. This is first cleavage, okay? And then second cell stage, then the fourth cell stage. The multiple cleavage occurs. I am coming to this. Repeated mitotic divisions in the zygote result in the formation of 2, 4 and 8 and 6 daughter cells. These daughter cells are called blastomeres. These daughter cells are called blastomeres. Okay, the embryo with 8 to 16 blastomeres are called the morula. It is important. The morula continues to divide and changes into the blastocyst while it moves further into the uterus. The blastomeres in the blastocyst are arranged in two layers. The outer layer is called trophoblast and the inner group of cells is called the inner cell mass. The trophoblast gets attached to the endometrium of the uterus and the inner cell mass gets differentiated as the embryo. After the attachment, the uterine cells divide rapidly and cover the blastocyst. As a result, the blastocysts become embedded in the endometrium. This is called implantation and it leads to the pregnancy. So, after the day 1, fertilization occurs, then first cleavage, in the day 2, the second cell stage, and day 3 to 4, the fourth cell stage, and this is uncompact morula, and then 8 cell morula occurs in the day 5, then early blastocyst in the late stage blastocyst hatching, and the implantation of the blastocyst at the last. There are another picture also, you may also refer to this. And in humans, the initial stage of pregnancy is referred to as implantation in short. So here are again various images, same images, but for your better understanding, I have provided this from various sources. Okay. Then this is called zona pellucida, and this is tropoectoderm, and this is blastocell, this is inner cell mass. In this picture, it is very much clear what I was talking about. This is epiblast and this is hypoblast. Okay, this is zona, this is inner cell mass. It is also good. So thank you. Hope you enjoyed this video. Please like this video and share among your friends and study hard. And bye bye.